All right, students, in this video, you're going to learn about transcription and translation, and also I'm going to provide you with guidance of how to do the gizmos. Now, transcription and translation is related to the central dogma of biology. So let me look up here. This is the central dogma of biology. The ultimate truth, the most important concept is that DNA is transcribed to RNA and RNA makes proteins. And proteins are responsible for most of the functions that occur in living organisms. So it's a very important concept to understand. Just a quick refresher, proteins have many functions. Here are some of those functions. They support the regulation, expression of DNA, RNA. They maintain your body. They provide you with all of these supports, antibodies. So they're really important, and you have to understand this complicated process. So hopefully you did the reading and the video. So these are our family, our Brazilian family again, albinism children. Remember, there's 25% chance of having an albino child if the parents are heterozygous for it. So the process that DNA, that the central dogma is about is protein synthesis. So let me read to you the definition. It says, protein synthesis is the process in which cells make proteins. It occurs in two stages, transcription and translation. Transcription is the genetic transfer of genetic instructions in DNA to mRNA in the nucleus. So kind of complicated, lots of terms, but after this, you're going to learn exactly what that means. So just really quick, this is more information that you need. The mutation that makes albinism is located on the 11th chromosome. This is the 11th chromosome, and this is the location where the mutated genes that cause our albino children to be albino. That's more information that you need, but it's interesting to be aware of how it's related to chromosomes and protein synthesis. So in this example, this is a complicated chemical pathway. You don't need to know this. I just wanted to show you. But this is how enzymes, which are proteins, they create products. The enzymes are a protein that speed up chemical reactions. They make things happen in the body. And the enzyme responsible for having melanin are all these different enzymes that are labeled in yellow. So you don't need to know this. All you need to know is that enzymes are proteins that make things happen in the body. For example, they make the body produce melanin out of a substance called tyrosine. That's all you need to know. Okay, don't freak out. All right. So these are the steps: transcription, and transcription is basically, you can think of this analogy, this understanding. And I found this picture, which is really cool. So imagine that you go to the library, you get a textbook, and you actually have to make a copy of the information in that textbook because you want to take that information to somewhere else. That's what transcription is, and that's what exactly occurs in the nucleus of a cell. So you're basically taking the information from DNA and you're making a copy of it in a special kind of molecule called RNA. There is a difference between DNA and RNA. DNA is two strands, double helix that are bonded together by the bases. These are the bases cytosine, guanine, adenine, thymine, the ATCG that you should know about by now. But RNA is different. RNA is a single strand, and we have cytosine, guanine, adenine. But now we have a new base, which is called uracil. Now, why is it different? It's just a biological fact. That's what RNA has. It has a different base. So what you're going to do first in the gizmo is you're going to transcribe DNA. You're going to take the information from DNA, that ATCG, and you're going to make a copy of that in the form of RNA. That's transcription. DNA, there's a copy of it, and it makes a strand of RNA. Then we'll get into translation later, but for right now, first step is transcription. So here's an example of transcription. We have our genetic code right here, our GAG well, I can't see, CACGTA, all these letters. And what you have to do in the gizmo, you have to transcribe it. And it follows rules. And the rules are right here. Is adenine complements uracil. When the gizmo asks you the complementary strand, it's asking what base will pair with the base in DNA. So, for example, let's do one together. So you'll learn that DNA. I don't know why that pops up. DNA is it comes in three. They're called codons, which we'll talk about later. But let's say with ATC, that is our DNA. But you want to copy that information to make messenger RNA. So what's going to happen is A is going to pair with U, T is going to pair with A, and C is going to pair with G. These are what I just did. I transcribed three bases from DNA to RNA. Let me make it bigger. Okay, so I want you to pay attention. CAC, the copy of the information would be GUG. That is the complement to that DNA information. So that's what you have to understand. DNA is the library, and the RNA is the copy of the book that you did, and you're going to take that information outside and use it to make something. In this case, the body is going to make that RNA into proteins, but that's translation. So in the gizmos, you're going to do that. You're going to answer these questions. It says, suppose you want to build a house. How would you communicate your design plans with the construction crew that would work on the house? Well, that's your answer. It's what you have to think. Um, Cells build large complicated molecules such as proteins. What do you think? Why do you think cells use as their design plans for proteins? What do you think the cell does? What does it use? So you do these questions. This is a double-stranded. What is that? What double-stranded molecule with ACGT? What is that? So you're going to answer the questions right here. You're only going to do activity A. Once you're done with activity A, do not turn it in. We're going to do this for this week and next week. So you're going to answer the question here. Which RNA base bonded with thymine? You have to figure that out by using the gizmo. So here's the gizmo. Let me reset it. How long is this video? Too long. No, it's not. Okay, so right here we have our DNA molecule, and you are going to use an enzyme. Remember, enzymes are molecules that make things happen, and you are going to make RNA. So you release the enzyme. There's the enzyme. Look, it's happening. Something's happening. So what you have to do is you have to pair. You remember you're making RNA, so it's going to follow different rules. So you're going to pair them just like that, like you've been doing. A with T and so on. So it says use the complementary nucleotides to create a strand of RNA. So remember A with T, and then, well, but remember, this is the DNA, right? DNA to RNA, A pairs with a new special nucleotide right here. So I'm going to give you a chance to mess around with it, but you are constructing an mRNA molecule from DNA. Remember, you're making a copy of the information here into messenger RNA. So in order to be successful for this assignment, you have to watch the video and use the notes. That's and ask questions. And then what you're going to do is you're going to answer these questions right here, and you're going to do a complementary base strand, and then answer the questions and stop here. Stop. Do not do activity B. Well, if you want, you can try, but it's not required for right now. So yes, that is what you're going to be doing Very important process. That process is happening right now in your body. Your body is making proteins to do anything. So hopefully that was clear, and you will be successful in this. Um, and that's it.